Good evening, everyone. Welcome to Sanhita Chatterjee's biology classes. In today's class, I will discuss about digestion and absorption of carbohydrates. Carbohydrates are the major macronutrients that are essential for production of energy. We can't do any work without energy. We all know that. And carbohydrates and proteins are known as energy giving food. Okay, and digestion means splitting of large food particles into smaller droplets. So here we will study splitting of carbohydrates. Digestion of carbohydrates. Firstly, carbohydrate digestion is a multi-step enzymatic process. Multi-step means that there is a several step include the processing of digestion that converts complex carbohydrates into simple sugar. Now why complex? Because there is a chain of sugar molecule present, because there are different class of carbohydrates. We can classify carbohydrates as per its sugar molecule, presence of its sugar molecule. If one sugar molecule is present, then it's monosaccharide. If two, then disaccharide. And if 3 are more than 2, then it will be called as polysaccharide. If we are having a chain of carbohydrates, here lots of sugar molecule is present. Sugar is the unit of carbohydrates. A single molecule is called as sugar. Okay, this is a unit of carbohydrate, unit of carbs. Simplest form of carbohydrate is sugar. So sugar is for absorption, because digestion is the process of splitting this food into the smaller particles into its simplest form. But after splitting it, what we have to do? These small particles must mixed with blood for its absorption, for its utilization, so that it can provide energy. Okay, so it involves mechanical breakdown, enzymatic hydrolysis, absorption in different parts of the digestive tract. You can see here a person is yellow color. Who is that? It's enzyme. Now who is the enzyme? What is the function of enzyme? This person is having a specific site for a substrate. This is a substrate. Okay, if it is a carbohydrate digesting enzyme, then only carbohydrates can bind with it, and after binding with this, if this substance bind with that place, that enzyme will split it out into the smaller droplets. So it's the enzyme and it's the active site. Oral phase, and that is the initiation of digestion. Firstly, carbohydrate digestion takes place at the mouth. Only carbohydrate digestion can occur within the mouth, because carbohydrate digestion enzyme is present at the mouth. Firstly, before the digestion starts within mouth, where is the mechanical digestion? Who performed mechanical digestion? Mechanical digestion is performed by our teeth. By chewing breaks food into smaller pieces, increasing surface area for enzyme. Then starts enzymatic digestion. Salivary amylase is the first enzyme who starts digestion of carbohydrates. And where do we get this salivary amylase? Salivary amylase is present within the mouth. In the mouth, we are having several glands like parotid gland, sublingual gland and submandibular gland. All these three types of gland produces salivary amylase. Salivary amylase is an enzyme that can split carbohydrates, but it's also having some characteristics that I will discuss one by one. Firstly, salivary amylase starts breaking down starch. What is starch? It's nothing but polysaccharides. Now poly means so many and saccharide means sugar molecule. If so many sugar molecule is attached with together, then it is called as polysaccharide. I have already made a video on carbohydrate biochemistry. You can see that, so that you will get idea of polysaccharides, monosaccharide, disaccharides and their principles, their nature, their structure, their properties, everything. Okay, I will attach that link into my comment box so that you can get that video and clear your views. So these polysaccharides firstly split it into two forms. First is maltose and second one is dextrins. So firstly starch. Starch is splitted out into maltose and dextrins. Maltose is a disaccharide and dextrin I will discuss later. So salivary amylase functions best at a neutral pH and is inactivated, point to be noted, it's get inactivated, it's lose its function by stomach acid, which is HCl. Stomach produces HCl and this acidic pH is not suitable for salivary amylase. Okay, so when food reaches the stomach, they certainly stops breaking down of carbohydrates. Okay, now let's discuss about salivary amylase. There is two type of starch. Starch means polysaccharide. Starch means polysaccharide and starch is of two types. First type is amylose. And second type is amylopectin. Point to be noted, starch is of two types, amylose and amylopectin. Now what is the difference between these two compounds? In amylose the sugar molecules attached in a straight chain. See, this molecule straightly attached with this. Okay, they are forming straight bond and that bond is named as glycosidic bond. That bond is named as alpha-1,4 glycosidic linkage. This bond is named as alpha-1,4 glycosidic bond. But in case of amylopectin, here is the straight chain and a branch is present. Here is the difference, is like a branch is present here, right? And this branch is formed by alpha-1,6 glycosidic bond. So alpha-1,4 glycosidic bond is present within amylose and alpha-1,4 and alpha-1,6 bonds are present within amylopectin. Salivary amylase only split alpha-1,4 glycosidic bond. Salivary amylase only breaks this bond, alpha-1,4 glycosidic bond. It cannot break alpha-1,6 glycosidic bond. Never ever. This bond cannot be broken down by salivary amylase. Salivary amylase only splits these bonds. So if these bonds get splitted, what type of molecule will be generated? Like this, maltose and maltotriose. Okay, amylose is formed with alpha-1,4 glycosidic bond. It will also produces two sugar molecules attached like disaccharide and it's the trisaccharide. 3 sugar molecule is present. But when we cleave this bond amylopectin, then we will get some alpha limit dextrin. So what is the difference between dextrin? Within dextrin there is presence of alpha-1,4 glycosidic bond and alpha-1,6 glycosidic bond. Alpha-1,6 glycosidic bond remain intact within dextrin. It cannot be splitted by salivary amylase. What is this orange color molecule particle? This is This one. This one is salivary amylase. So salivary amylase only split into this bond. Okay. And so we get segments of two molecules, two sugar molecules, that is maltose, and three sugar molecule compound like maltotriose. Like here is the long chain of carbohydrates. This one. It's a long chain of carbohydrates, and this is named as polysaccharide. This is named as polysaccharide. 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 And they all are attached with alpha 1,4 glycosidic bond. Alpha 1,4 glycosidic bond. And this molecule is named as amylose. But in case of amylopectin, here is presence of this branch. And this branch is formed by this bond. Here is this bond, that bond, that bond. These bonds are alpha 1,6 glycosidic bond that can't cleave by salivary amylase. Okay. So salivary amylase only split alpha 1,4 glycosidic bond, and we get disaccharides, maltose and dextrine after cleaving this alpha 1,4 glycosidic bond. Now we reach the second phase of carbohydrate digestion, which is phase 2, and that is named as gastric phase, that occurs within stomach. Actually, carbohydrate digestion starts within mouth, then the food particles after splitting move towards the stomach, and when it reaches towards stomach houses some cells, specific cells that are parietal cell. And what is the function of parietal cell? So parietal cell secretes an acid that is HCl, hydrochloric acid, and the pH gets changed. Firstly it was alkaline pH, now it's acidic pH. Okay, so in acidic pH, that salivary amylase get inactivated. Salivary amylase get inactivated. The acidic pH of the stomach denatures salivary amylase. Denature means it cleaves or it destroys the salivary amylase. That what? That acidic pH, and stopping carbohydrate digestion temporarily. Salivary amylase splits that starch, and starch is of two types, amylose and amylopectin. Within amylose there is only alpha 1,4 glycosidic bond is present, and within amylopectin alpha 1,4 and alpha 1,6 both glycosidic bond is present. So after splitting this, but salivary amylase only split that alpha 1,4 glycosidic bond, and alpha 1,6 glycosidic bond remain intact. So the compound in which alpha 1,6 bond is present, that is dextrine, right? Dextrine is famous for having alpha 1,6 bond, and another we get maltose, which is a disaccharide. Okay. Third phase of carbohydrate digestion is intestinal phase. So within mouth we are having food without separating the particles. Firstly, that I have already discussed, that we are having pizza, we are having rice, but we are not separating the carbohydrates, that now we are only having carbohydrates, now we are only having protein. We do not eat like this manner, we eat whole food together, right? So that is why it is mixed with starch, lactose, sucrose, all things are together. Now this salivary amylase is secreted from the glands, parotid gland, sublingual and submandibular glands. After that, salivary amylase split these all things, the alpha 1,4 glycosidic bond, and produce sucrose, lactose and also alpha limit dextrins, where alpha 1,6 bond is present. Okay. Next digestion occurs before small intestine. There is another person, that is pancreas. Yes, pancreatic amylase. Pancreas also having pancreatic amylase. Here is also another amylase. And amylase function is same. It only speeds alpha 1 4 glycosidic bond. So here we also getting disaccharides. Okay, so see, pancreatic amylase action. The pancreas secretes pancreatic amylase into the duodenum. This enzyme continues breaking down starch into maltose, disaccharide, maltotriose, trisaccharide, and dextrine, where alpha 1 6 glycosidic bond is present. And for maltose and maltotriose, there is only alpha 1 4 glycosidic bond. Okay, and within dextrine, there is alpha 1 6 and alpha 1 4. Both bond are present. But alpha 1 6 is unbreakable by salivary amylase and pancreatic amylase. So for digestion of dextrine, we have to reach small intestine. Last phase of the carbohydrate digestion takes place within the small intestine. Why? Because small intestine houses some enzymes that are responsible for breakdown of the last products of carbohydrates and produce the simplest form, that is glucose. Okay, so where the enzymes are present at the brush border area. And what are brush border enzymes? Brush border enzymes are digestive enzymes that are found on the microvilli of the small intestine's epithelial cell. So small intestine having epithelial cell and epithelial cell having microvilli. This is the structure of villi, not microvilli. This whole structure, that is a structure of villi, which is a finger-like projection, and that is a prime characteristics of small intestine. Okay, and small within villi there is epithelial cells. These cells are epithelial cells, and these epithelial cells houses these microvilli that are shown in this picture but not clear. Okay, so what I am trying to say, that there is a structure like that these cells is epithelial cell, and on the surface of epithelial cell there are so many dedicated hair like projections that are named as microvilli, and microvilli houses that brush border enzymes. Now what are those enzymes? Basically, these enzymes are maltase. Now the name of the enzyme, write it down: maltase, sucrase, and lactase. These three enzymes. Maltase, lastly ase means enzyme. Maltase, sucrase, lactase. These three enzymes that will act on which type of food? Maltase will break down maltose. Maltase will break down maltose and produce maltose. If you split one molecule of maltose, maltose is a disaccharide. That means there is two sugar molecule. One is glucose, another is glucose. So two molecule of glucose will be produced if you break one molecule of maltose. One molecule of maltose if you break, then you will get two molecule of glucose. Why? Because one molecule of maltose houses two molecule of glucose. That is why maltose is a disaccharide, and the enzyme that split maltose is maltase, and that is a brush border enzyme. Next enzyme is sucrase, and its substrate is sucrose. It will act on sucrose, and sucrose is a byproduct of glucose and fructose. So if you break sucrose, you will get one molecule of glucose and one molecule of fructose. One molecule of glucose and one molecule of fructose. And the last, lactase. Lactase will work on lactose, and it will split it into glucose and galactose. One molecule of glucose and one molecule of galactose. One molecule of galactose. Okay, this classification again you will get it the whole story in my last in my another video that is based on carbohydrate biochemistry. Okay, there you will get all this information. So maltase will break down maltose into two molecule of glucose, sucrase will break down into sucrose and will produce glucose and fructose, lactase will act on lactose and produce glucose and galactose. Okay, so and lastly again we are having two A's, that is dextrinase and glucoamylase. Dextrinase and glucoamylase, these two are the specific enzyme. Dextrinase will break down dextrine, that is unbreakable throughout the stages of pancreatic amylase, right? So from mouth to pancreas there is no breakdown of alpha 1,6 bond. We have to wait until the food particle reach into the small intestine. There you will get dextrinase and glucoamylase enzyme that can split the alpha 1,6 bond and further break down dextrins into glucose. Okay, so here is the summary. Let's discuss the digestion of carbohydrates under one roof. Firstly, we will see that carbohydrate digestion starts within mouth. So here is the mouth, and from mouth that salivary amylase secreted, and its specificity that it only cleaves alpha 1,4 glycosidic bond, so that it will produce maltose is a disaccharide and dextrine. The specificity of dextrine is alpha 1,6 glycosidic bond is present, and maltose it's a disaccharide. Next, move to the stomach. So from mouth it moves to the stomach. So this is the stomach, and stomach houses the cells named as parietal cell. Parietal cell secretes HCl. Now what is the function of HCl? HCl, salivary amylase, in presence of HCl it gets inactivated. That minus red sign means salivary amylase lose its power. Okay, so it gets denatured, it gets distracted by the presence of acidic pH. HCl means that pH gets acidic. Okay, next, that pancreas, it's pancreas, and pancreas also secrete pancreatic amylase. Now amylase means it will only cleave alpha 1,4 glycosidic bond, and by cleaving alpha 1,4 glycosidic bond it will also produce disaccharide. Di means 2 and saccharide means di means 2 and saccharide means sugar. So 2 molecule of sugar. Okay, disaccharide and trisaccharides. Next, move to the small intestine. This cell is a small intestine. Within small intestine, the main specificity of small intestine is villi is present within the small intestine, and within villi there is epithelial cells, and epithelial cells houses microvilli, enterocyte. So this cell is named as enterocyte, that is present within small intestine. And enterocyte houses some brush border enzymes. So what are the brush border enzymes? Brush border enzymes are, first brush border enzyme is lactase. Second brush border enzyme is maltase. Third brush border enzyme is sucrase. And last one, most important one, is isomaltase. Isomaltase means dextrinase or glucoamylase, also named as isomaltase. Okay, so what they do? Lactase splits lactose. Lactose is a disaccharide, so it will split that bond. Lactose will split that bond. That bond means this bond. Okay, and it will produce two molecules. Lactose will produce two molecules. One is glucose, this one is glucose, and another one is galactose. Okay, maltose, this will also split two and produce two molecule of glucose. Two molecule of both are glucose and glucose. Okay, next here this one is sucrose. So sucrose will also split this 1,4 glycosidic bond, produce two molecules of, one is glucose and another one is fructose. Okay, but what is the function of isomaltase? Isomaltase perform a very important function, that it splits the alpha 1,4 and alpha 1,6 glycosidic linkage. Alpha 1,4 and alpha 1,6 glycosidic linkage. It will split this bond alpha 1,6 and also this bond alpha 1,4. Okay, and will produce 4 molecules of glucose. Glucose, 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 all are glucose. Okay, so after digestion, what type of molecules we get? Basically these all are monosaccharide, only one sugar molecule, but so many type of monosaccharides. What are they? First one is glucose, second one is fructose type, and third one is galactose. Basically these three types of monosaccharide we get after digestion of carbohydrates. Okay, so what are these? They are glucose, glucose, next one is galactose, galactose, and last one is fructose. Absorption mechanism. Absorption mechanism also takes place within the small intestine. Which cell? Enterocytes. So enterocytes are the cells of small intestine. Okay, enterocytes of the vilus, and enterocyte is again present within the vilus. Okay, so vilus is present within the small intestine because vilus are the special characteristics of small intestine. Now small intestine houses vilus and vilus houses enterocytes, and within the enterocytes there occurs absorption of carbohydrates. After digestion of polysaccharide, what are the product we get? We get glucose and galactose from lactose. If we split one molecule of lactose by the enzyme lactase, we will get glucose and galactose. Next is the fructose. Fructose is present within the sucrose. One molecule of sucrose gets splitted into one molecule of fructose and one molecule of glucose. And lastly is maltose. If we break maltose, we will get two molecule of glucose. So these three important monosaccharides that we get after digestion, that are glucose, galactose and fructose mainly. So these three things have to transport where they are still present at the brush border site of the small intestine. So it has to cross the small intestine and then it will mixed with the blood, and that blood will be carried out by the portal vein and then it will reach to the liver. Okay, so for our travel, we always use for traveling some transporters like bus, train, aeroplanes, right? So for cell, for transporting glucose, galactose and fructose, they also use transporters. So what are the transporters? There are mainly three type of transporter. First one is SGLT1. Write it down into your copy, SGLT1. Now S for sodium, G for glucose, sodium, glucose. These two things are transported together. That is why co-transporter. Co means when two molecules transport together. Anything that is carried out by two things, that are regarded as a co. Okay, so coordinators, co-transporters, like that. So sodium with glucose, that means glucose cannot enter into the enterocyte alone. It need the help of sodium. That is why it takes the help of sodium to enter into the enterocyte. I will discuss this process, but firstly just understand that glucose use a transporter to enter into the enterocyte, that is named as SGLT1. That means glucose sodium glucose co-transporters, SGLT1, and this transportation need to use energy. Okay, like that, we need tickets for our traveling. Like this transporters also, if this glucose using these transporters, then it also has to pay some energy. That is why these transporters are named as active transport, because it requires ATP. Without money it won't transport. Okay, and for money, cell is having only one type of money, that is ATP. That is ATP. Okay, next, for fructose, fructose is very special. Fructose will not share any transport system that is shared by glucose or galactose. It will be transported separately. For transportation of fructose into the enterocyte, it need a specific type of transporter, that is GLUT5. So always remember, GLUT5 is only specific for fructose. Okay. So, first one is SGLT1. SGLT1 is used by glucose as well as galactose. Glucose and galactose both can enter through this SGLT1, but they have to go with sodium. If glucose is going, it will go with sodium. If galactose is going, it will go with sodium. So, sodium glucose transporter will transport glucose and galactose together. But for fructose, there is a special type of transporter named GLUT5. And after, when they reach within the enterocyte, fructose, glucose and galactose, then these three things have to transport to liver. To reach liver, they use a same transporter, that is GLUT2. Okay. So see, next is the blood transport. All monosaccharides enter the hepatic portal vein and travel to the liver. The liver converts fructose, galactose into glucose. Okay. So how this process occurs? Again, we already studied that there is a digestion that occurs firstly within mouth, then it stops within stomach, then it again happen within pancreas, and lastly within small intestine. So this is this site, that site is the site of brush border site, this site. Okay. So within brush border, there is also some brush border enzymes that produces some end products like glucose, galactose and fructose. Now it has to be transported within the small intestine, that is enterocyte. Okay, then it has to cross enterocyte, and there comes the another place, that is blood, and from blood they have to take up and go through this portal vein to the liver. This one is portal vein. Okay, so how this transport process occurs? Firstly, there occurs an energy generation process, constantly occurring generation of energy. How? By the help of sodium potassium ATPase pump. There is a pump that is named as sodium potassium ATPase pump. What it do? It actually removes sodium out of the cell and enter potassium within the cell. Okay, 3 sodium get out and 2 potassium get in. So there is within the cell, sodium is less within the cell. This is a cell, this is enterocyte, and within the cell Na plus is less within the cell because sodium is moving out and potassium is moving inside of the cell. So to maintain this equilibrium, to maintain the continuous supply of sodium, we have to send sodium into the cell. That is why we need a transporter that is named as SGLT1. This is named as SGLT1. And what is the function of SGLT1? SGLT1 means it transport sodium with glucose. So sodium will go inside. Sodium will go inside, so that inside sodium concentration get higher. because here it is the outside of the cell. Here is the sodium concentration is so high. Within the outside of the cell, sodium concentration is very high. So sodium concentration is very high. So sodium enter from the outside of the cell into the enterocyte with glucose and galactose. With glucose and galactose. This one is glucose, this one is galactose. They are also going inward. Glucose and galactose, they both enter with sodium. That is why SGLT co-transporter, and they are using this pump using ATP, which is produced by sodium potassium ATPase pump. This is named as sodium potassium ATPase pump. This pump is responsible for generation of ATP, and that ATP is used by SGLT1. SGLT1 used that energy, and SGLT1 transport sodium with glucose and galactose. Okay, is it clear? Sodium from the outside to inside by the SGLT1. Glucose and galactose can enter within the small intestine. First one. Second one is the fructose. But fructose will move separately. For fructose, there is a separate type of pump is present here. What is that? That is named as GLUT5. That is named as GLUT5. GLUT5 only transport. This one is GLUT5. This GLUT5 is only responsible for fructose to move inside the enterocyte. Okay, so GLUT5 SGLT1 is complete. Next, there is GLUT2. This transporter is GLUT2. This transporter will transport three particles. First one is glucose. First one is glucose. Second one is glucose. Okay. Second one is galactose. And third one is fructose. These three particles will move through this GLUT2. Okay. These three particles will move through this GLUT2 into the blood. And then there will be glucose, there will be galactose, and there will be fructose. They all will be uptake they all will go through this portal vein and reach to the liver for its utilization. Okay. So, with a quick recap, I will tell you the whole summary. Firstly, what happens? See here. Within the mouth, there is salivary amylase, and it splits the alpha-1,4 glycosidic bond into maltose and dextrin. Dextrin is still having alpha-1,6 glycosidic bond intact. Then these whole things shift toward the stomach. In presence of stomach HCl, they lose their property. Salivary amylase get inactivated and carbohydrate digestion partially stops at the stomach. Next, the food particle move to the pancreas. Pancreas secrete pancreatic amylase, and this pancreatic amylase also able to break the alpha-1,4 glycosidic bond, and they will also produce disaccharide and trisaccharides. Next, we reach the small intestine where there is villus, and within villus there is a cell that is enterocyte. And enterocyte houses three types of enzyme. First one is lactase, second one is maltase, and third one is sucrase. And fourth one is isomaltase. Isomaltase is very important because it is similar like the enzyme dextrinase and glucoamylase. These two enzymes is able to split the alpha-1,6 glycosidic bond. So what happens? Okay, lactase splits lactose and produce one molecule of glucose and one molecule of galactose. Next, maltase splits maltose and it convert it to two molecule of glucose. And sucrose, it gets splitted by sucrase and produce one molecule of fructose and one molecule of glucose. Okay, and then isomaltase splits this alpha limit dextrine and we produce that glucose only. So there is 3 types of monosaccharide after the completion of digestion. That is glucose, next is the galactose, and another one is fructose. And these all things, these all 3 monosaccharide has to be transported into the enterocyte so that it can reach from the enterocyte to the portal vein and then ultimately to the liver. Okay, so here within the enterocyte there is another energy generation pump, that is sodium potassium ATPase pump. It always remove 3 sodium out of the cell and incorporate 2 potassium into the cell. That is why sodium concentration is very less within the cell. To maintain this sodium balance, at the brush border site there is a lots of sodium available. So sodium move into the enterocyte by SGLT1, sodium glucose transporter 1. With sodium it is transporting glucose and galactose. Okay, next, fructose is very special. For fructose we need a separate pathway, separate private car, that is GLUT5. So fructose only pass through transport through GLUT5 into the enterocyte. And the last thing, that after reaching into the enterocyte, glucose, galactose, fructose, all 3 things will pass through GLUT2 to reach the blood. And from the blood they are taken up by the portal vein and sent to the liver for its utilization and energy production. Okay, hope you understand these things well. And there is some important picture. See, this is an enterocyte and at the brush border site there is SGLT1 one transporter, GLUT5 second transporter, and GLUT2 third transporter. There is another transporter missing, that is sodium potassium ATPase pump. SGLT1, through SGLT1 glucose and galactose both can enter into the enterocyte. GLUT5, it is only specific for fructose. Okay, glucose transporter 5, that is why it is named as GLUT5. SGLT1, its full form is sodium glucose co-transporter 1, and lastly GLUT2, that is glucose, galactose and fructose. All 3 things will pass through from the enterocyte to the blood capillary through the GLUT2. Okay, important characteristics of the transporters. The absorption of carbohydrates in small intestine involves several key transport proteins. First one is SGLT. We already know this name, sodium glucose link transporter. Okay, SGLT1. Next is GLUT. We have 2 type of GLUT, GLUT2 and GLUT2 and GLUT5. Right, GLUT2 and GLUT5. Next, ATPase pump, sodium potassium ATPase pump. Okay, so first thing is SGLT, that means sodium glucose link transporter. Its location, SGLT1 is primarily located on the apical membrane of the enterocytes. That means it is located on the surface of the enterocyte. You remember that there is a cell, that is the enterocyte, and here is the surface of the enterocyte, that is the surface, and that membrane is named as apical membrane. This is the apical membrane. Okay, this is the apical membrane site. Next, mechanism is very important. Transporter uses sodium gradient to absorb glucose and galactose from the lumen of the small intestine into the enterocyte. Another important thing, there is a new word, that lumen of small intestine. It's very easy. What is called lumen? If this things is an enterocyte, okay, so outside of the enterocyte is lumen. This place is lumen, and this is the enterocyte. This is inside of the cell, this is outside of the cell. Outside of the cell is named as lumen. Okay, within the lumen there is lots and lots of sodium available, right? And within the enterocyte there is very less amount of sodium available because of the sodium potassium ATPase pump. So see, SGLT1 is a secondary active transport system. Secondary, why secondary? Because they use the energy, that ATP that is already generated by sodium potassium ATPase pump. It relies on the energy generated by the sodium potassium ATPase pump. Okay, it is dependent. Which one? That SGLT1 is dependent on sodium potassium ATPase pump. That is why they are named as secondary active transport system. For every glucose molecule absorbed, 2 sodium ions are co-transported into the cell. So with glucose, 2 sodium ion co-transported into the cell. The sodium ions then diffuse down their concentration gradient, providing the energy to bring glucose into the cell. Next is the GLUT. GLUT location. GLUT transporter, especially GLUT2, are located on the basolateral membrane. What is the basolateral membrane? The side of the enterocyte facing the bloodstream. Okay, so here I will draw again that if it is a enterocyte, then it is having two sides. First one, this side is apical site, apical site, and that side is basolateral side, because here is the blood, right? Here is the blood and here is the intestinal tract. So within the intestinal side, that will be the apical side, and within the blood, that will be the basolateral membrane side. Okay. Next, once glucose enters the enterocyte via SGLT1, it need to be transported into the bloodstream for utilization. This is where GLUT2 comes into play. GLUT2 is a facilitated diffusion transporter. Facilitated means that it do not use energy. It gets facilitated. Okay. So, meaning it does not require ATP. It allows glucose to pass down its concentration gradient from the enterocyte into the blood. And last type is the ATPase pump. This pump is located on the basolateral membrane of the enterocyte. Okay. So, basolateral membrane, you know that if it is an enterocyte, then this part is the basolateral side. Okay, here is the location of sodium potassium ATPase pump. Next, the sodium potassium ATPase pump plays a critical role in maintaining the sodium gradient. It actively pumps sodium out of the enterocyte and potassium into the cell, using ATP to do so. This creates a low sodium concentration inside the cell and a high sodium concentration outside the cell. This gradient is essential for the function of SGLT1 because it allows sodium to flow back into the cell and bringing glucose along with it. Okay. And lastly, the fifth step, that is metabolic utilization and storage. We have already studied digestion and absorption, and after digestion and absorption there comes a very important step, that is metabolic utilization and storage. So why do we require absorption? Because of the production of energy, utilization of these products. So immediate energy use. Cells use glucose for ATP production through glycolysis. This is a process by which carbohydrate generate ATP. Okay, and glycogenesis, it is a storage. Excess glucose is stored in the liver and muscles as glycogen. And lipogenesis means fat storage. If glycogen stores are full, extra glucose is converted into fatty acids and stored in adipose tissue. That means if we take a lot of carbohydrate that we required normally, so more carbohydrate will store into the adipose tissue and it will increase our fat deposition within the body. So intake of lot of carbohydrate is not good for our health. Okay, so carbohydrate intake is equal to weight gain. That should not be done. Okay, that is the conclusion. So here is the whole process. Hope you have enjoyed the class, and if you want me to share more videos like this, then stay connected. Thank you.